Hello everyone. Today I'm going to be talking about my research on using a novel circular convolution-based association method for identifying genetic correlations between pre-existing medical conditions and COVID-19. My name is Wang and I'm from Shanghai American School. So firstly, I would like to give a brief overview of how this presentation will be structured. First, I'll give a background to understanding my research, then how I filtered my data, my methodologies I used, the results I obtained, and finally the conclusions I had for my research. So first of all, the background for my research project. As you probably know, COVID-19 is a pandemic that has raged all around the globe. It has left nearly 2.4 million dead, and this high lethal rate is probably due to COVID-19's high transmission rate, as well as the slow showing and sporadic symptoms. And so over the past year, doctors and physicians have noticed an interesting trend, and that is how pre-existing conditions, such as chronic kidney disease, can lead to a more severe case of COVID. This trend or pattern is more often known as comorbidity patterns, which is essentially the presence of one disease leads to a more severe case of another disease, which in this case is a more severe case of COVID-19. Now that we know this, it is important to highlight that observing this trend has only been observed on the phenotypical level. Essentially, if doctors and physicians were to use these patterns and trends to help predict individuals' COVID severity case, they can only do this by looking at that patient's past phenotypical medical records. So in my research, I plan to extend this observation into the genetic level, essentially identifying the genetic genes that drive this comorbidity pattern right now only observed on the phenotypical level. So to get to that level, first of all, we have to understand how this comorbidity trends occur on the genetic level. So hypothetically speaking, let's have a COVID-19 patient and a coronary artery disease patient. And so here we have a chunk of the human genome. On the top is how we would expect a normal person's genome to look like, AGGG, etc. And on the bottom is essentially what we realized to be a reoccurring theme for COVID-19 patients. So essentially, we've realized that for a majority of COVID-19 patients who have a more severe case of COVID, they have a reoccurring theme of a mutation at this position from G to C. We have the same thing for coronary artery disease. Keep in mind that this is the same chunk of human DNA across these two diseases. So the expected here is the same strand of DNA. So now we highlight the mutations or single nucleotide polymorphism, which is just a fancy word for mutations on a single nucleotide, on a single position. We highlight these SNPs. This is the key here. And so, and it is important to know that these are all reoccurring mutations and SNPs. They're not just sporadic ones. And so the reason why there are comorbidity trends between coronary artery disease and COVID-19 is because of an overlapping reoccurring SNP across the two diseases. So again, let's re-emphasize that the disease-associated SNPs, or just reoccurring SNPs for a particular disease that occur on the same positions, are drivers of comorbidity patterns observed phenotypically. And now you might wonder, how exactly do we recognize these reoccurring disease-associated SNPs? How do you differentiate that with a sort of a sporadic mutation on a randomly selected on a person at a random spot? So essentially, how we do that is to use the power of the scientific community. There are studies called genome-wide association studies, which target and which has a goal of trying to find these disease-associated SNPs for a particular disease. And so now that we have the background covered, let's move on to how I filtered my data in my project. So first, I downloaded these GWAS study results online, and which should be in this format. So one row is the positions of these mutations, SNPs, and the other row is p-values. The p-values for a particular SNP is essentially a measure of how closely that SNP correlates to the occurrence of a particular disease. So essentially the probability or the correlation score. And so with this data I downloaded from the internet, I then used the QQ plot to identify the significant SNPs and filter out the insignificant ones. So a significant SNP means a very low p-value, which means a higher association with that disease. So this is a very detailed explanation of how I filtered my SNP. It's through a standard process called QQ plots. So first I took the negative log value of the p-values I obtained online, and I also took a negative log of essentially the p-values that I would expect if all of these SNPs were insignificant. And so from that, let's zoom in on this place. We see that sort of what we have and what we expect deviates at 1.0, meaning that so essentially I would filter out all the SNPs below this threshold of 1.0 because that's what we expect and that's not what we want to analyze, and only take the SNPs that deviates from our expected distribution of SNPs. Now that we have filtered these SNPs, this slide essentially shows the general structure of my project. So I downloaded 6 comorbid disease GWAS study data. I downloaded the COVID-19 GWAS study data, and I first filtered out the insignificant SNPs for all 7 of these diseases, and I used a circular convolution-based associate algorithm to sort of find the to process essentially COVID-19 and these 6 comorbid diseases by pairing them up with each other, and I obtained the results in. Okay, so now it's time to discuss essentially again what constitutes as the same SNP. So we have this diagram from a couple slides before, and now let's take away these other SNPs for now and only focus on this set of SNP. So right now they're on the same position. So we're very confident that sort of this SNP is a driver for comorbidity patterns because they're on the same position. However, what if this SNP is one nucleotide or one base pair on the left of the other SNP, or if it's one on the right or 2 on the right? It's important to note that the human genome is billions of nucleotides long, so 1 or 2 nucleotides away from each other isn't really that big of a difference. So essentially, we have to figure out a way to account for this sort of deviation or this minor differences in the distance between 2 SNPs. And so how I was able to do that is through a thing called haplotype blocks. So haplotype blocks are evolutionary conserved blocks where SNPs occur in clusters. So essentially they're genetically defined. So right now we again have the previous diagram. Now we have another set of data, the p-values for each one of these SNP mutations obtained through GWAS studies. And now we add in the borders for each haplotype block. These are downloaded online and they're predefined such that all the SNPs occurring in, for example, haplotype block 2 occur in a single cluster, occur through clusters. And now moving forwards, essentially in my study, I no longer make associations between the 2 diseases on the single SNP level. I no longer do that. Now I make associations and find associations across these 2 diseases on the entire haplotype block level. So exactly how I do that, I'm going to show you on the next slide. So essentially, I'm going to use a circular convolution algorithm to find the association score of these 2 diseases on a single haplotype block. How I do that is first I define I find the score of each of a single pair. For example, a single pair of SNP. For example, the 0.02 SNP in COVID and 0.24 SNP in CAD, which is coronary artery disease. So I find the score of this pair using this equation. I plug in the p-values, I multiply them together, and I find the difference in their distance, and I take the inverse of that. And with this, I obtain a pair score of sort of a score for this pair of SNPs. Then to get the score of the entire, the association score, the hap score of the entire haplotype block, I then essentially, I I take the combination of each one of the SNPs in the COVID to each one of the SNPs in CAD, and for each line, I get the pair score, and I add all the scores of each one of these lines together to finally result in essentially the score for the entire half-life block, the HAP score. And so because I'm multiplying these together and adding them together, this is the reason why I'm calling this algorithm a convolution. And since I'm exhausting every single combination of these pairs, it's also the reason why I'm calling this a circular convolution algorithm. Before we move on, I would like to put emphasis on how I used the max function here. So essentially, the reason why I use the max function is because if there is sort of two SMPs, this pair that occur on the exact same position, then the difference of their positions would be 0. And with 0 on the denominator, the pair score would be close to infinity, and we don't really want that. So so essentially we set the max we set the difference to be at most 1, so it would never reach 0, so we never have that problem of having infinite number of pair scores. Now, essentially, this is how we get the score of one half-life block across by comparing two diseases. So I would get the HAP score for all the other half-life blocks, and I would essentially find the half-life blocks with the highest HAP scores, and these half-life blocks would essentially contain the genes that drive the comorbidity trends between the two diseases. And so one half-life block that I realized that essentially occurred that had high values across all of these diseases was half-life block number 3 in chromosome 6. And so essentially, I realized that this particular half-life block had the highest HAP score in lung cancer, meaning that it probably has the highest correlation with lung cancer has the highest correlation in this half-life block with COVID. And in this half-life block, there's a gene called KHDRBS2. This gene is an RNA binding domain. And so an interesting thing I found online at NCBI with KHDRBS2 is on this graph here. This graph here shows the gene expression for this particular gene. And from here, we see 3 sort of peaks. One is at the lung. So in the lung, there's a high gene expression of KHDRBS2, which matches our sort of data of how the half-life block containing this gene has the highest HAP score when it comes to lung cancer. Another interesting thing that validates sort of my finding for this gene is the fact that it also has a very high expression in regards to the human thyroid. So as we all know, thyroid is a very crucial sort of symptom for COVID infections. So this interesting correlation, in a way, validates my finding of this particular gene as a potential driver for comorbidity patterns. Another interesting thing is how this gene is highly expressed in the brain, which potentially may lead to more future research on whether or not certain brain diseases has an association and can lead to a more severe case of COVID-19. So this is a sort of direction for future research. It can be. Another half-life block that I found that had a very, very high HAP score of this huge number with regards to chronic kidney disease is number 243 in chromosome 3. And so in this half-life block, there's a prominent gene called ATR serine thiorein kinase. And this gene is primarily responsible for DNA damage sensor, activating cell cycle checkpoints. So with regards to this gene, another interesting thing that I found on NCBI and also published on PubMed is that this particular gene is very highly correlated with breast cancer. And breast cancer, just how happens, it's on the CDC's list of existing health conditions that will increase your chances of hospitalization upon getting infected by COVID-19. So this is another very interesting validation of my finding of this particular gene, such that this gene is a driver for chronic kidney disease and COVID-19, and it's also a big driver for the occurrence of breast cancer, which just so happens is also a comorbidity disease with COVID-19. This is a very interesting finding. Another finding I realized, so after I realized that breast cancer has a correlation with chronic kidney disease, I analyzed breast cancer and I found this gene that drives that's a very big driver for the comorbidity patterns between breast cancer and COVID-19. And this gene, it just so happens, is also transcriptionally activated in lung cancer, which is also a pre-existing condition on the CDC website for COVID-19. So it's very, very fascinating to see how all these different comorbidity diseases, when you go into the genetic level, uh, some of them are sort of clashing, have the same genetic drivers as each other, which is very cool and very interesting. These are some of the other genes I realized I have found, and I'm not going to go into very much detail in this presentation. And so, to conclude my experiment and my research, the significance of what I did is that I was able to find the underlying genes that drove comorbidity patterns between pre-existing conditions and COVID-19. And so, by finding these genes, I'm able to provide a foundation for the future where there are maybe gene-based COVID-19 symptom severity diagnosis, which would allow for the influence of comorbid diseases to be accounted for even in carrier patients. So there may be patients who don't physically or phenotypically express symptoms for lung cancer, but they have the mutations that may lead to lung cancer and leads to the comorbidity patterns. So even if this patient they don't express lung cancer phenotypically, I'm still able to find this comorbidity pattern and help account this into the severity diagnosis algorithm, which will increase the accuracy of the diagnosis because it's based off of genetics now, not only phenotypical medical records. In addition, another significance of my study is that it sets the precedence for a method of using a haploid block-based approach to identify comorbidity driving genes. And by identifying these genes, they can be used to find other comorbid diseases, such as breast cancer in my research, or even the brain disease, the brain disease, which can sort of spark future research and help us better understand the disease. So thank you for listening. I know this is a bit over the time limit, and this is a chunk of my bibliography. If you want to see the full bibliography, please look at the online shared document to see the full poster. Thank you so much, and thank you.